Thank you for attending our webinar, A Guide to Immuno-PCR. Our speaker today is Emma Easthope, Technical Content Writer at Innova Biosciences. Emma has over 15 years experience of working within the life sciences industry, including companies such as AstraZeneca, Millennium Pharmaceuticals and Novus, before she joined Innova in early 2015. She has extensive laboratory experience within various immunodetection techniques. In the presentation today, Emma will describe how Immuno-PCR draws on the advantages of both ELISA and real-time PCR. She'll go through the main steps of an Immuno-PCR assay and introduce you to our ThunderLink Plus technology, a quick and easy method of conjugating antibodies to oligonucleotides, making Immuno-PCR considerably more accessible as a method of immunodetection. And there will be a Q&A session at the end of the presentation, so please submit your questions by typing in the questions box in the control panel. We will answer as many questions as possible within the time we have, and any remaining questions will be answered by email following the presentation. So now let's welcome Emma.
Thanks, Lucy. Okay, first let's begin by talking briefly about immunodetection. The term immunodetection describes the process of using antibodies to detect antigens in a sample. Well-established immunodetection techniques include Western blotting, immunocytochemistry, immunohistochemistry, flow cytometry, and ELISA. Since immunodetection relies on the use of antibodies, these methods are sensitive, highly selective, and applicable to the detection of a wide variety of analytes. Although the immunodetection methods mentioned on the previous slide have been around for decades, Immuno-PCR is a relative newcomer. It was first developed in 1992 and combines the antibody-based specificity of an ELISA with the amplification of PCR to provide extremely high levels of sensitivity. To fully understand the Immuno-PCR process, we'll first take a closer look at ELISA and PCR. The well-known term ELISA is an abbreviation of enzyme-linked immunosorbent assay. The technique was invented in the 1970s and evolved from the radioimmunoassay methods which were developed during the previous decade. While radioimmunoassays require the use of radioactive labels, an ELISA is a much safer form of immunodetection. As the name suggests, the ELISA readout relies on an antibody which has been linked to an enzyme such as horseradish peroxidase, alkaline phosphatase, or glucose oxidase. In the presence of an appropriate substrate, the enzymatic activity produces a colour change, and this can be measured. As assay techniques have evolved, alternative readouts have been developed. The term ELISA is now broadly used to describe any assay where a molecule has been adsorbed onto a solid surface. In addition to being a much safer option than radioimmunoassays, ELISAs offer a number of further advantages. They're fairly simple to perform, and unlike techniques such as Western blotting or immunohistochemistry, produce a quantitative readout. They're also sensitive, providing analyte detection in the microgram to picogram range, and they're relatively inexpensive to run. Furthermore, they can be easily adapted to HTS formats and used to screen large numbers of compounds. The disadvantages of ELISA are less obvious. However, if the ultimate aim of the assay is to evaluate a parameter such as the tissue distribution or the molecular weight of the analyte, it would be wise to select an alternative method. If the ELISA is reliant on a labelled secondary antibody for detection, this can be a source of contaminating background signal. Finally, colorimetric ELISA readouts are not amenable to multiplexing, although this can be overcome through the use of fluorometric readouts. A number of different ELISA formats are commonly employed. An antigen-down ELISA requires the antigen to be attached to a microtiter plate, as shown in the left-hand panel. Indirect detection of the antigen relies on the binding of an antigen-specific primary antibody, followed by the binding of a labelled anti-species detection antibody. In this example, the detection antibody has been labelled with an enzyme, which produces a colour change by acting on an appropriate substrate. Direct detection of the antigen requires the use of a labelled primary antibody. This can save time by reducing the number of steps which are performed in the ELISA, and it can also eliminate the non-specific signal which can be introduced through the use of secondary antibodies. During a sandwich ELISA, a capture antibody is bound to the microtiter plate, as shown in the middle panel. The antigen is then added, followed by a second antigen-specific antibody. This type of assay can also use either indirect or direct detection. Indirect detection uses a labelled secondary antibody, while direct detection relies on a labelled primary antibody. This eliminates the need for the secondary antibody incubation step. Sandwich ELISAs are generally more sensitive than antigen-down ELISAs. A competitive ELISA is most commonly used when the analyte is small and cannot be bound by two different antibodies simultaneously, or in cases when only one antibody specific to the antigen is available. There are a number of different ways in which a competitive ELISA can be formatted. In this example, in the panel on the right, the antigen has been attached to a microtiter plate. If the same antigen is present in the test sample, there will be competition for binding to the directly labelled detection antibody. In this setup, a high concentration of antigen in the sample is associated with a low signal output. This is because the antigen in the sample will bind the detection antibody, which will then be washed away during the course of the ELISA, rather than becoming bound to the surface of the plate. As already mentioned, it's often beneficial to use a directly labelled antibody for detection, rather than relying on the use of secondary antibodies. Lightning Link from Innova Biosciences enables quick and easy labelling of antibodies, proteins, peptides, or any other biomolecule which contains free amine groups. These labelled reagents can then be used in a wide variety of techniques, including ELISA. The Lightning Link reaction is initiated simply by adding the biomolecule into a vial of the lyophilized mixture which contains the label of interest, and incubating. With no separation steps, there's no loss of precious reagent. We offer a wide range of Lightning Link labels, including enzymes, fluorescent dyes, fluorescent proteins, biotin, and streptavidin. You might also be interested to know that we've recently launched our Europium conjugation kit, enabling you to conjugate your biomolecule to specially treated 200 nanometer europium particles. Antibody europium conjugates are ideal for ELISA assays, which rely on time-resolved fluorescence. Why not visit our website to find out more? The polymerase chain reaction, PCR, was invented by Dr. Carey Mullis in 1983, a discovery for which he was subsequently awarded a Nobel Prize. PCR is a technique that is used to amplify a specific segment of DNA from only a small amount of starting material, and can rapidly produce more than 1 million copies via a simple 3-step cycling process. The first step in this process involves heat-induced denaturation of double-stranded DNA to separate the two complementary strands. This is commonly performed at 94 or 95 degrees C for 30 seconds. Next, the temperature is lowered to allow binding of PCR primers. These serve as starting points for DNA synthesis. The choice of temperature for this step, which typically lasts for 30 seconds, is dependent on the melting temperature of the primers, and is usually between 50 and 60 degrees C. The final step in the PCR cycle is the extension step, during which a thermostable DNA polymerase produces a complementary copy of the target DNA strand beginning at the primer. This step is performed at the optimum temperature for the polymerase, typically 72 degrees C. The duration of this step is dependent on the length of the segment which is being amplified. It's usual to perform 25 to 35 rounds of this 3-step cycle. Too few cycles means that not enough product is generated for adequate detection, while too many cycles can result in the production of unwanted secondary products. The number of copies of the target DNA sequence which are formed after n cycles is defined by 2 to the power of n. Early methods of viewing the amplified DNA sequence relied on gel electrophoresis in polyacrylamide or agarose, followed by ethidium bromide staining. Ethidium bromide is a DNA intercalator, which inserts into the spaces between the DNA strands and allows the DNA to be visualized on a UV transilluminator. The size of the amplified sequence is determined by including a relevant marker on the gel and should be identical to that which was predicted from the known DNA sequence. Restriction enzymes, which digest at sites within the amplified sequence, can be used to provide further confirmation that the specific target has been amplified. The DNA fragments can also be assessed by Southern blotting, a process which requires the DNA to be transferred from the gel to a membrane. It's then detected by hybridization with labeled sequence-specific probes. Gel electrophoresis followed by ethidium bromide staining is an endpoint detection method. The PCR products are detected and characterized only after the reaction has finished. During real-time PCR, also known as quantitative PCR or qPCR, data is collected throughout the PCR process as it occurs by correlating production of the PCR product to fluorescence intensity. Two general types of fluorescent reporters are used: dyes which bind to double-stranded DNA and fluorescently labelled probes. Cybergreen dye and TACMAN probes are well-known examples of each of these. Cybergreen dye was developed as a safer alternative to ethidium bromide, which is known to be mutagenic. The free dye has only a low level of fluorescence, but upon binding to double-stranded DNA, the fluorescence is greatly increased. As more and more double-stranded DNA is produced, the fluorescent signal grows in intensity. TACMAN probes work by the principle of fluorescence resonance energy transfer, or FRET. A reporter fluorophore is attached at the 5' end of the TACMAN probe, while a quencher fluorophore is attached at the 3' end. These two fluorophores are therefore held in close proximity. Following excitation by a light source, the reporter fluorophore reaches an excited state. However, its fluorescent emission is suppressed by the quencher. TACMAN probes are designed to anneal to the specific DNA sequence, which is to be amplified during the PCR reaction. As the DNA polymerase carries out DNA synthesis, it uses its exonuclease activity to cleave nucleotides sequentially from the 5' end of the TACMAN probe. This causes separation of the reporter fluorophore from the quencher, giving rise to a measurable fluorescent signal. While cybergreen will bind to any double-stranded DNA molecule, the TACMAN assay is specific to a particular target. Labeled probes, such as the TACMAN probes, therefore provide the opportunity for multiplexing. Real-time PCR offers a number of clear advantages over traditional PCR. Firstly, since no post-reaction processing is required, it's significantly faster. It also provides increased sensitivity, as it can detect much smaller quantities of the starting material than traditional methods. In addition, real-time PCR affords a large dynamic range, significantly greater levels of sample throughput, and most importantly, it's quantitative rather than qualitative. The only clear disadvantage to real-time PCR is that it requires expensive instrumentation. However, its advantages far outweigh the disadvantages. The PCR reaction can be divided into 4 main phases: the linear ground phase, the early exponential phase, the exponential phase, and the plateau phase. During the linear ground phase, which usually lasts for the first 10 to 15 cycles of the PCR reaction, the fluorescence is below background levels. The baseline fluorescence is calculated during this phase. At the early exponential phase, the fluorescence is significantly higher than background levels. The cycle at which the fluorescence crosses a defined threshold, usually 10 times the standard deviation of the baseline fluorescence, is referred to as the cycle threshold, or CT. Amplification is optimal during the exponential phase, but as the reagents become limiting, the plateau phase is reached. Analysis of real-time PCR data relies on instrumentation. In the example shown here, the graph on the left shows PCR data from 6 samples, each of which contains a known starting amount of DNA. The red curve on the far left is derived from the sample with the highest amount of DNA, while the black curve on the far right corresponds to the sample with the lowest amount of DNA. The cycle number has been plotted against the level of fluorescence, and the CT value has been calculated for each sample. The lower the starting concentration of DNA, the higher the CT value. The instrument software has then been used to plot the initial quantity of DNA against the CT values. The CT values of samples of unknown DNA concentration can then be related back to this set of standards to allow the concentration to be determined. A wide range of real-time PCR instrumentation is commercially available. In addition to the price, factors which should be considered when selecting a suitable reader include its size, block format, and well capacity, all of which will dictate how many samples it can hold and will impact on the level of throughput. Some readers offer the option of individually programmed wells, which can be really useful when optimizing PCR conditions. Many instruments are provided with heated lids to prevent sample evaporation. The maximum ramp rate should also be considered. This is the maximum rate at which the heating block can change temperature. Faster ramp rates are more desirable, since they will considerably shorten the length of the entire PCR protocol. The instrument software should also be evaluated for user-friendliness, as well as for whether it can be integrated easily with other software or data handling packages which are in use in the laboratory. As mentioned before, Immuno-PCR brings together the methods of ELISA and real-time PCR to create an extremely powerful technique. Since an ELISA requires the use of antibodies, it's adaptable to any protein. However, its sensitivity is not adequate for the detection of analytes of low abundance. While real-time PCR provides exponential signal amplification, it can't be used directly for antigen detection. Immuno-PCR relies on the use of an antibody which has been conjugated to an oligo, and this conjugate acts as a bridge between the immunoreaction and DNA amplification. The technique of Immuno-PCR therefore delivers extremely high levels of both specificity and sensitivity. As with an ELISA, either an antigen-down assay or a sandwich assay format can be used. In this image, a sandwich assay is shown. A typical Immuno-PCR sandwich assay consists of a number of defined steps. Firstly, an antibody, which is specific to the target antigen, is immobilized on the surface of a microtiter plate. After washing to remove any unbound antibody, the plate is blocked, and then the sample is added. Further washing is used to remove unbound sample. Then a second specific antibody, which has been coupled to an oligonucleotide, is added. After a final set of washing steps, the DNA is amplified and detected via real-time PCR. Like any form of immunodetection, each step of an Immuno-PCR assay requires careful optimization. The aim of the optimization process is to maximize the specific readout while keeping the background signal to a minimum. To keep things simple, it's suggested that when developing an Immuno-PCR assay, the parameters of an existing immunoassay are, if possible, adapted and modified. Factors which should be taken into account include the plate coating process, the nature of the wash buffer and blocking solution, the sample incubation step, the plate layout, as well as the choice of relevant antibodies. These steps will now be discussed further. During the plate coating step, the capture antibody is bound to the microplate by non-specific adsorption. This is usually performed overnight at 4 degrees C. Typically, a carbonate, bicarbonate or borate buffer with a pH of greater than 9 is used to dilute the antibody for plate coating. In addition to the choice of buffer, the optimal concentration of the coating antibody should be determined. A range of 0.2 to 20 micrograms per ml is a good starting point, and it can often be a good idea to perform a grid experiment, since this allows multiple antibody concentrations to be tested simultaneously on the same plate. Once the capture antibody has been successfully coated onto the microplate, the plate should be washed to remove any unbound antibody. Typical wash buffers include phosphate-buffered saline or tris-buffered saline, often with the inclusion of a low concentration of detergent, such as tween 20. Efficient washing throughout the entire Immuno-PCR assay is essential to minimise non-specific binding. The number of duration of the washes should be carefully optimised. After coating the microplate with the capture antibody and washing, the well should be blocked to prevent any non-specific binding of the reagents and to maximise the signal-to-background ratio. Typical blocking buffers include weak solutions of proteins such as BSA, non-fat dried milk, gelatin, casein, or whole serum diluted in wash buffer. These passively adsorb to all of the microplate surfaces which aren't already occupied by the capture antibody. The choice of blocking reagent, the concentration of the blocking protein, and the duration of the blocking step should all be optimised. In some instances, it may be beneficial to block overnight at 4 degrees C. Since Immuno-PCR provides such high levels of sensitivity, test samples can often be considerably diluted. This significantly reduces any background effects that may be caused by the nature of the sample material. The binding of the capture antibodies to the antigen is a specific high-affinity interaction. Because of this, short incubation times are often sufficient for the sample incubation step, although this should of course be optimised during assay development. It's also important to determine an appropriate incubation temperature for this step. Any well-designed experiment will include relevant control samples to allow for the sensible interpretation of results. Positive controls will typically consist of samples which have been spiked with known concentrations of the antigen, while negative controls will consist of sample material which has no antigen present. Ideally, the controls should not all be contained on one side of the microplate, since this will mask any plate drift that can occur during the assay. The inclusion of a calibration curve is essential for calculating the antigen concentration of test samples. Since Immuno-PCR has such a broad dynamic range, this can be generated by using tenfold serial dilutions of the antigen. If you wish to reduce the size of the quantification window, the dilution steps can be lowered, for example to 5-fold or below. Ideally, all samples should be analysed in at least duplicate, and for consistency, it's essential that controls and standards be diluted in the same matrix as that of the unknown test samples. The success of any immunoassay is dependent on the quality of the reagents which are employed. Immuno-PCR relies on the use of highly specific antibodies for both capture and detection of the antigen. If either of these antibodies recognises a common molecular motif, it may bind to the same epitope on multiple targets, which will significantly increase the background signal of the assay, since proteins other than the target of interest will be detected. As well as being highly specific, it's important that the capture and detection antibodies should each target a different epitope. If both antibodies recognise the same epitope, binding of the capture antibody to the antigen will prevent the detection antibody from binding to the antigen, resulting in a low signal. Another consideration is whether to choose polyclonal or monoclonal antibodies. The performance of polyclonal antibodies varies from one immunised animal to another, and once it's been used up, a particular lot of a polyclonal antibody can't be reproduced. A permanent antibody supply therefore cannot be guaranteed. Furthermore, only a fraction of a polyclonal antibody preparation will consist of the antibody of interest, meaning that immunogen affinity purification is essential to enrich antibodies which demonstrate the required specificity. Monoclonal antibodies are often preferred since large quantities of a specific antibody which recognises a single antigenic epitope and demonstrates a uniform performance can be produced. Another obvious advantage of monoclonal antibodies is that they do not require immunogen affinity purification. As well as being adaptable to the detection of any protein and generating exponential signal amplification, Immuno-PCR offers a number of further advantages. It's extremely sensitive, providing analyte detection in the picogram to femtogram range, and is suitable for very small sample volumes. It's also compatible with complex samples such as serum or urine, not least because these can be diluted considerably prior to running the assay. Since Immuno-PCR requires fewer incubation steps than an ELISA, assay reproducibility is improved, and the time to results is significantly decreased. Immuno-PCR also has a much wider dynamic range than an ELISA, and it's highly amenable to multiplexing. By using specific antibodies against multiple targets and conjugating each of those antibodies to a different oligo, multiple readouts can be obtained simultaneously from the same precious sample material. One major disadvantage of Immuno-PCR is that it relies on the conjugation of an antibody to an oligo, a process which can be costly and time-consuming, and which requires expert knowledge. To overcome the issues that beset traditional methods of antibody-oligo conjugation, we've developed our ThunderLink Plus oligo-conjugation system. This facilitates the quick and easy production of antibody-oligo conjugates. Our ThunderLink Plus oligo-conjugation system is an easy-to-use kit which enables stable antibody-oligo conjugates to be generated quickly and efficiently and requires no specialist knowledge for use. The oligo is added to a vial of oligo activation reagent and the antibody to a vial of antibody activation reagent. Then both vials are incubated for 30 minutes at room temperature. During the incubation step, 2 desalt columns are washed. The activated oligo mix is passed through one of the columns, and the activated antibody mix is passed through the other. The antibody and the oligo are next incubated together for 1 hour at room temperature, and the conjugate is then ready to use. An optional cleanup step can be performed at this point to remove any unbound oligo, should this be necessary. ThunderLink Plus offers a wide range of advantages over traditional methods of antibody-oligo conjugation. In addition to being quick and easy to use, the kit provides high levels of antibody and oligo recovery. The bond which is formed between the antibody and the oligo is covalent, so the resulting conjugates are extremely stable. And because the chemistry is unidirectional, there's no risk of cross-linking. So only antibody-oligo conjugates are formed, no antibody-antibody or oligo-oligo conjugates are produced. The ThunderLink Plus technology is flexible, allowing you to use your own antibody and oligo at your chosen ratio. And since the linking chemistry works at either end of the oligo, there's the possibility to add extra modifications. As well as including an optional post-conjugation cleanup step to remove unbound oligo, the kit is provided with a positive control antibody and oligo to confirm that the conjugation has been successful. Furthermore, the ThunderLink Plus kit is freeze-dried, so it ships at ambient temperature and has a long shelf life. And of course, like all of our products, ThunderLink Plus is stringently QC tested, providing you with consistent high quality and excellent batch-to-batch reproducibility. The ThunderLink Plus oligoconjugation system is designed for the activation of 100 micrograms of antibody in 100 microliters of a suitable buffer. The antibody should be purified and at a concentration of 1 mg per ml, while the buffer should have a pH of between 7 and 9, should be amine-free, and if sodium azide, BSA or gelatin are present, these should be at concentrations of below 0.1%. Full buffer considerations are provided with the protocol, as illustrated. For antibodies which do not meet these criteria, we offer our AbSelect range of antibody purification kits. These allow a simple buffer exchange to be performed, enable concentration of the antibody, or provide the option of antibody purification from complex solutions such as tissue culture supernatant, ascites fluid, or serum. The Absolute kits are easy to use, provide high levels of antibody recovery, and require no specialist knowledge or equipment. ThunderLink Plus is intended for the activation of an oligo that is HPLC purified, has a concentration of 60 to 100 micromolar, and which is supplied in at least 100 microliters of a suitable buffer. Full buffer considerations are again found within the protocol. ThunderLink Plus can be used to conjugate both single and double-stranded oligos. Single-stranded oligos should be of 10 to 120 bases in length and should contain a terminal amine group at either the 5' or the 3' end. This should be added during synthesis. Double-stranded oligos should be of up to 80 base pairs in length, but only one of the strands should be terminally aminated. Since the ThunderLink Plus chemistry works at either the 5' or the 3' end of the oligo, the technology is compatible with modified oligos, such as those which have been biotinylated or labelled with a fluorescent dye. We've found the conjugation efficiency of ThunderLink Plus to be slightly higher with 5' aminated oligos. Following activation and desalting of the antibody and the oligo, both reagents are ready for conjugation to one another. ThunderLink Plus can be used to generate conjugates with a range of different antibody to oligo ratios, which can be achieved by simply adding varying amounts of oligo to the antibody. It should be noted that the antibody to oligo ratio will only ever be an average, since a population of labelled antibodies will be produced during the conjugation reaction. Each antibody within this population won't necessarily have the same number of oligos bound to it. A 1-to-1 ratio is highly likely to result in some unbound antibody being present at the end of the reaction. The overall conjugation efficiency is typically 95 to 100%, and as we mentioned before, it's marginally higher when using oligos which are aminated at the 5' end. Innova Biosciences offers a wide range of custom services. When it comes to Immuno-PCR, our experienced scientists can help you to optimize your antibody-oligo conjugate and can also produce your chosen conjugate at a scale to meet the requirements of your assay. The performance of an antibody-oligo conjugate is often related to the number of oligos which are bound to the antibody. Our antibody-oligo micro-optimization service is a fast and efficient way to optimize your conjugate using only very small quantities of your antibody. This service provides you with 6 different conjugates, which include different chemistries and different antibody to oligo ratios. You can test these in your chosen application to identify the one which demonstrates optimal performance. Our custom antibody oligo conjugation service follows on from our micro-optimization, or can be used as a standalone option. This provides you with your chosen conjugate at a scale from 1 mg to 1 gram. Both services include antibody cleanup prior to conjugation, removal of unbound oligo following conjugation, and proof of successful conjugation in the form of a QC gel. Why not have a look at the custom services video on our website to find out more? We hope this webinar has given you a good general overview of Immuno-PCR, as well as letting you know how Innova Biosciences can help develop your own Immuno-PCR assay. Immuno-PCR is an extremely powerful technique. It combines the specificity of an ELISA with the amplification of PCR. It's adaptable to the detection of any protein and provides robust and reproducible data in only a short timescale. Furthermore, it's highly amenable to multiplexing, saving you time, money, and precious sample material. Although Immuno-PCR isn't yet the #1 go-to method of immunodetection, it's rapidly grown in popularity. The number of publications citing its use is growing steadily. Our ThunderLink Plus technology enables the conjugation of an antibody to an oligo in just a few simple steps, making Immuno-PCR considerably more accessible to the wider scientific community. Thanks for your time, and we'll now take any questions.
Okay, thank you for your presentation, Emma. So it's now time for the Q&A session. I can see that we have some questions coming in already, as discussed earlier. If we don't get around to answering all of your questions now, we will be sure to get back to you via email after the webinar. So let's get started. Okay, so how can I confirm that my antibody to oligoconjugation has been successful?
So the best way of doing this is to use the conjugate in your downstream application, but it's also possible to analyse your conjugate by gel electrophoresis. So if you run a small amount of the conjugate on a reducing SDS page gel and then stain that for protein, you should be able to see the heavy and light chains of the antibody, and those will demonstrate a band shift when they're bound to the oligo. Obviously, the size of the band shift will be dependent on the size of the oligo.
Okay, so once the antibody and oligo have been activated, you say that they require desalting. Where can I purchase desalting columns and how do they work?
Actually, our ThunderLink Plus oligoconjugation kit is provided with all the reagents that you need, and that includes the desalting columns. So how they work is by gravity flow. They're packed with Cephros beads which contain pores. Large molecules such as antibodies or oligos will pass through the column relatively quickly since they won't get trapped by the pores, and therefore they'll exit the column before any smaller molecules such as salts.
Okay, thank you. And here's another one. Are you able to supply me with the oligos?
Yes, we can do that as a custom service, which will be subject to a small admin fee. Just get in touch if you want to know about that.
Okay, great. And can I use ThunderLink Plus to conjugate an antibody to RNA?
Unfortunately not, because the reagents which we provide in the ThunderLink Plus kit aren't RNase free, so the kit's actually unsuitable for antibody RNA conjugation. We can perform this service for you as custom work. Just get in touch if you need further information.
Okay. And then we've got time for a couple more. So how much more sensitive is Immuno-PCR than ELISA?
The limit of detection of an ELISA is usually within the microgram to picogram range, whereas Immuno-PCR offers detection in the picogram to femtogram range, so it's much more sensitive. The limit of detection in an Immuno-PCR assay will vary depending on the choice of antibody, but it can usually detect the antigen at around 1,000-fold lower concentration than that which would be detected by an ELISA against the same target.
Okay, thank you. And then the last question, can I use my antibody oligoconjugate for a technique other than Immuno-PCR?
Yes, you definitely can. Antibody oligoconjugates are ideal for Immuno-PCR, but they also are regularly used for assays such as proximity ligation or proximity extension. Those techniques rely on the use of two oligoconjugated antibodies against different epitopes of the same protein, or against two biomolecules which can interact with each other. So when both antibodies are bound to their targets and they're close together, the oligos hybridize and you can then amplify the DNA and detect it.
Okay, thanks Emma. So that's all we have time for today, unfortunately, and we'll get back to you with any questions that we haven't yet answered. Thank you for joining us for today's webinar, and thank you Emma for your presentation. Please don't hesitate to get in touch if you want to know any more about anything we have presented today, or if you would like to know more about our ThunderLink Plus oligoconjugation kits, or our custom labelling, optimization or bulk conjugation services. We have an expert technical support team here ready to answer any questions that you might have. You can also follow us on Facebook, Twitter and LinkedIn to follow regular updates here at Innova, including upcoming conferences we'll be attending, future webinars, application notes and more. Thank you, and we hope to see you at our next webinar.